Hello everyone, how are you? Welcome back. Hope you had a good week. So most of the people who usually come are here. Let me just give everyone another minute or so before we get started. How has your revision been? How has the week been? Let me know how you guys are doing. Hope you're not feeling too stressed. I know it's coming up pretty soon now, next month. Just a reminder, we've got today's session. We haven't got a session next week. It's having a break after 4 weeks, and then we're going to restart the week after next. So we've done these 4 sessions, 1 week off, and then we've got the last 4 sessions, and that will bring us right up to the exam date. So just a reminder about that. And yeah, let me know how you guys are doing, how everything has been. Are you managing to get much done on the question banks, or people started using mocks or anything like that? Just let me know how things have been. Okay, I think we'll get started. So today we're doing the other half of the neuroscience, basic neurosciences. So we touched on some of the content last week, and now we're going to do the rest of it. As always, so I've just tried to keep it focused on the essential things. There is a lot of content, and it's impossible to kind of get through every single part of it, but these are the things that I found most important, and the things that come up in questions, both in the question banks and also in the real thing. So obviously we'll focus on those, and I'll try to keep it close to syllabus. So if you have a syllabus with you, then that will always be helpful. So we're going to have a look at some of the neural circuits, and as I'll go into in a moment, we've actually covered some of them already, and we're going to cover some of them in a different kind of way later on. Some of the modulators, neurodevelopment and neuroplasticity, and also neurodegeneration. And as I mentioned before, we've got no session next week. We'll resume the week after next, and that will be on molecular genetics. So after that, we will have completed 3 whole sections of the syllabus. So that will be quite good. We would have done 3 sections, and we would have just not too much left really, part 4 and 5. So the pharmacology stuff, and then the classification. So obviously those 2 are quite big, but yeah, it's going well. You guys have covered a lot. So let's have a look at neural circuits. So in the syllabus, it's got a lot of information in terms of things that we need to look at, and we actually, as I mentioned, have covered some of these already in different aspects. So things like appetite, I have put in a couple of the same slides from last session just to as a reminder, but also things like sleep, that's covered in different places, in parts, sort of, it's all a bit mixed together. And again, things like aggression, we covered some of that in the first session, and other parts of it kind of interlink with each other. So if it looks like this kind of thing isn't covered as much, it's because most of it is touched on in a different way. So usually if we're going through things like ADHD or like in classification, we will be looking at the mechanism of how these things work. So that will be why it will make more sense when you're doing the topic. So as a reminder, so we had a look before at how the hypothalamus is involved in appetite, hunger and thirst. So the kind of physiological mechanisms by which those things are regulated in the body. So the hypothalamus has neurons which sense nutritional and hormonal signals from the blood. And it increases appetite via these various mechanisms when there's kind of low glucose. So it has these special neurons that detect blood glucose, and it will increase appetite when it finds that there's a low glucose level. And those are found mainly in the arcuate nucleus. The area posterema also senses appetite hormones, and you may remember that that's one of the circumventricular organs, so where the blood-brain barrier is a bit more permeable. So just looking at this diagram, yeah, as you can see, like from the stomach, you've got things coming up. So, well, actually start with the sandwich. So the person's eating the sandwich, goes into the stomach. From the stomach, you've got the short-term satiety signals, and then you've got the blood glucose that comes up, and that's looked at by the hypothalamus. You've also got other things going on in the long term. So you've got the leptin, you've got the insulin, and then also the other things that regulate kind of satiety, like when you've eaten enough, those are all coming out. So that cholecystokinin, neuropeptide Y, and those sorts of things. So it's all kind of linked in together. And just as a reminder, the same from last week's session, which was these hormones of appetite. So cholecystokinin, which reduces appetite, which is secreted by the duodenum and jejunum in contact with food. It stimulates gallbladder and it stimulates the pancreas, and again it acts on vagal neurons to signal satiety to the medulla, and also that kind of goes to the hypothalamus as well, in terms of the satiety signals. And then ghrelin, that acts directly on the hypothalamus, and then leptin as well, and then neuropeptide Y is actually made in the hypothalamus and is a response to sort of not having a low blood sugar, so that would increase appetite. Then the other ones are somatostatin, which is secreted by glands in the stomach, which inhibit the release of gastrin and HCl, inhibit secretin, and inhibit the release of glucagon as well. And then secretin is secreted by duodenal cells, and stimulates the pancreas to neutralize all of the things that have come out. And then gastrin stimulates exocrine cells to secrete HCl and pepsin. There's quite a lot to remember there, but again, just quite worthwhile in terms of EMQs. They're quite easy marks if you just try and remember what their functions are, but obviously there's a lot to cover, and I completely understand like that there's just so many things to learn, so this is just another one of those. This is something that is interesting in that it's in the syllabus, but it doesn't often come up in some of the study kind of materials that we use commonly for paper A. But this is the default mode network. So this is interesting because it's a network that's activated when you're remembering things, but also thinking about the future and then imagining things in the future. And the hypothesis is that the default mode network facilitates mental models and simulations. It involves the prefrontal cortex, the ventromedial prefrontal cortex, and the dorsomedial prefrontal cortex, as well as the insular, the inferior parietal lobule, lateral temporal cortex, and the hippocampal formation. So I think it's just useful for, I mean, it's sometimes an exam question, but it's just a hypothesis for things about imagining upcoming events and thinking about the future. So just worthwhile remembering that for that reason. And then the other thing is the sleep stages, which does come up in different areas as well. So it comes up in the in human development as well as in classification when we come on to sleep disorders. But basically, with the sleep stages, you've got your rapid eye movement, which is your REM, and your non-REM. So typically, non-REM is divided then into those 4 stages, which you can see. So going from awake, you go down into stage 1, stage 2, stage 3, and then stage 4, which is the deepest sleep, and then come back to REM, and then go back down, and that typically happens around sort of 3, 4, 5 times a night, depending, and lasts around 90 minutes in total for each sleep cycle. And yeah, so those are the waves that are involved. So stage 1, you've got theta waves, hypnic jerks, which is the kind of myoclonic jerks of when you're falling asleep and feel like you're falling off a building or something. And then stage 2 is classically is the sleep spindles. So those are only found in stage 2 sleep. That's a common question. Also K complexes. So K complexes and sleep spindles found in stage 2. And at this point, this is the kind of more physiological changes as well, the drop in temperature, and yes, drop in temperature, drop in rest rate and heart rate as well. And stage 3 delta waves. At this point, it's very hard to wake someone up. And then again, in stage 4, there's high dream activity as well. So there's dreaming in REM as well as in stage 4. So they're both hypothesized to have different mechanisms and reasons why there's dreaming in both of those, but they are not the same kind of mechanism for sleep, for dreams, sorry. So during an average night's sleep, most people spend about 25% in REM and 75% in non-REM as well. And as I said, it's about 90 minutes for each sleep cycle. So just moving on to some of the modulators. So in the syllabus, it's classified as all of these kind of neuroendocrine things, hypothalamic and pituitary hormones. Again, this is something that comes up in different sections as well, going over things, but it is fairly common to be asked maybe 1 or 2 questions on this. So this is looking at the HPA axis, so the feedback loop between the hypothalamus and pituitary and adrenal glands. What's most important about these is the kind of the link between mental illness, which I'll go into, but during stress, the hypothalamus secretes the corticotrophin releasing hormone and the other hormones, which can basically increase the adrenals secreting cortisol. And having that increase in cortisol obviously affects stress level, but also it has different long-term effects as well on the body. And also the amygdala and subgenual ACC are overactive in depression as well, and the dorsolateral prefrontal cortex is generally underactive. So this is why it's important, because most of the questions that will be asked will focus on this kind of theory that normally when you give someone dexamethasone, cortisol levels are supposed to decrease in the dexamethasone suppression test. However, in depression and also in some of the other psychiatric illnesses, the cortisol levels do not decrease, which basically gives this idea that there's a different mechanism for cortisol when it comes to mental illness. And in the glucocorticoid receptor hypothesis of depression, the dysfunction of this axis is linked to things like genetic defects or acquired illness. So what they're trying to say is that there may be a different expression of glucocorticoid receptors in people with psychiatric conditions. And for example, tricyclic antidepressants have worked on glucocorticoid receptors in the past, whereas that is not the case for SSRIs. So there are basically multiple methods of making changes to the kind of overall mood of a person, but that may not be just purely through SSRIs or serotonin. It can also be through this glucocorticoid receptor as well. And then it's also been found that early trauma can lead to HPA axis dysfunction, which may suggest a predisposition to depression. And I think also, like, obviously with chronic stress and things nowadays, like, it's a lot more common for people to develop depression. So there may be some link to that as well, which is not necessarily genetic or anything, could just be environmental as well. So if you're looking at the diagram, you've got this kind of chronic stress, the effects on the hypothalamus and the pituitary gland, causing sort of increased ACTH and affecting the adrenal glands. And that kind of affects all these other things as well. So resistance to glucocorticoids, increasing the inflammatory cytokines, which was also interesting because it's been found that there's a lot of inflammation in some of the depressive disorders, and this could be a possible reason why that is. And then HPA axis is hyperactive in major depression, which we have mentioned, but there's also adrenocortical enlargement. So there's actually a physical change that can be seen. And there's also increased corticotrophin releasing hormone and other things which are found in other parts of the body, including in the CSF. So just having a look at that diagram is basically saying that with chronic stress, there's increased chronic levels of cortisol, which causes prefrontal cortex degradation, amygdala hyperactivity, and could potentially lead to depression. So yeah, as said before, depressed patients do not show the normal level of cortisol secretion induced by giving like dexamethasone, the synthetic corticosteroid. So that's in about 50% of depressed patients. And yeah, so that's probably the most important part of this whole section. And then looking at bipolar disorder in particular, the corticotrophin releasing hormone does change in terms of levels just prior to hypomanic and manic episodes. That has been seen in research. And so you probably will know that, you know, there's the changes in the circadian rhythm, like people not sleeping, and all of those things kind of have a more organic feel to them. And this is part of the reason why. So some of the HPA axis changes. So in patients with bipolar disorder, you can get this limbic hypothalamic pituitary axis stimulation, which reduces pituitary volume and leads to pituitary hyperactivity as well. And the amount of endocrine changes that are noted in a patient does relate to how severe the manic episode can be as well. So now we're going to have a look at some of the hypothalamic nuclei. So I know there's loads of them and it looks like a lot of stuff, but it is a lot of stuff, excuse me. And I think it's just like one of those things that comes up in questions again that we just have to learn. So I've just put them separated them into kind of sections that maybe make a bit more sense together. But again, there's no real way of there's not really anything to understand as such. It's more just learning them. So the ventromedial nucleus is involved in body weight regulation, insulin regulation, sexual behavior and appetite, and lesions to the ventromedial nucleus can cause hyperphagia, so increased eating. The lateral nucleus also does body weight regulation and appetite, but lesions of this cause anorexia, anorexia being loss of appetite, not the condition anorexia nervosa. And stimulation of the lateral nucleus causes an increase in appetite. Then in the dorsomedial nucleus, again, this relates to body weight regulation and appetite. This stimulates your appetite and is also leptin sensitive, so sensitive to leptin in the blood, which would come from the adipose tissue as a satiety hormone, and then also manages libido, circadian activity, and anger, which is interesting. So there's an emotion of rage is also partly modulated by dorsomedial nucleus, and stimulation of this nucleus causes obesity and aggressiveness. So these three, ventromedial, lateral, and dorsomedial, just remember they're a lot to do with the body weight, to do with eating, to do with appetite. Then these anterior, posterior, and arcuate. So the anterior nucleus is involved in thermoregulation. It regulates the parasympathetic system to help keep the body temperature cool. So there's these neurons. Basically, there's these neurons that sense the temperature and they activate the parasympathetic system to help keep the body temperature down, and lesions to this cause hyperthermia. So if you're unable to regulate your temperature to decrease it, that causes hyperthermia. And then the posterior one also does thermoregulation, and that helps to keep the person warm, and lesions to this cause hypothermia. So if you just remember that anterior helps keep you cool. So maybe I like to think, as a bit of a random thing, but you're sitting in the car, you're sitting at the front, so the AC is on you and you feel more cool. So sitting at the front, anterior, the anterior keeps you cool. and maybe you might get a bit more hot in the back of the car, I don't know. And then the arcuate nucleus, it does also do body weight regulation. It's leptin sensitive as well, does appetite, releases dopamine, and secretes growth hormone. It secretes growth hormone releasing hormone to the anterior pituitary. And this one, lesions in here lead to amenorrhea and galactorrhea. Then these are the last four of the nuclei of the hypothalamus. So the paraventricular, this secretes corticotropin releasing hormone and the thyroid version of that, and then regulates ADH, regulates oxytocin, and lesions in the paraventricular nucleus cause diabetes insipidus. Supraoptic nucleus produces oxytocin and ADH, and lesions again cause diabetes insipidus. This is because of the ADH. And then preoptic nucleus stimulates LH and FSH from the anterior pituitary via gonadotrophin releasing hormone, which is kind of like pulsating hormone, which causes those sexual hormones to be properly regulated. Then thermoregulation and sexual behavior. And interestingly, lesions prior to puberty cause delayed sexual or arrested development in puberty. And then lesions after puberty cause impotence or amenorrhea. So basically affects the sex hormones. So if the person hasn't gone through puberty yet, it will kind of delay the whole thing. Or if they have already, it will stop whatever has occurred already. And then the last one is the suprachiasmatic nucleus, which is involved in circadian rhythm, and lesions lead to sleep disorders. So I think we basically covered most of the important ones from that. I don't think it's necessarily important to know exactly where each of them are, but just a good idea to know about them. And then I'm just going to recap the ones that are most important. So again, as I said, the suprachiasmatic nucleus, that is the body clock, that is the innate kind of thing that drives the circadian rhythm or circadian rhythm, and there is an intrinsic rhythm in those neurons. It does relate to other nuclei, so the suprachiasmatic nucleus is activated by light, and then that activates the paraventricular nucleus, and then that activates the pineal gland to stop melatonin coming out. So that's why they say like it's important to get sunlight on your face, or go outside, get sunlight on your eyes, because basically affects the nuclei and inhibits the melatonin release, so allows the circadian rhythm to be properly managed and give you a normal body clock. And a recap of the thermoregulatory nuclei. So the anterior, posterior, and preoptic nuclei are the most important ones for thermoregulation. So as said, anterior hypothalamic nuclei is sensitive to warmth. So if you get too hot and you're sitting in the front of the car, you turn the AC on and then you become cold. So the anterior hypothalamic nuclei activates parasympathetic system to cause heat loss or to cool you down. And then when you're too cold, the posterior hypothalamic nuclei, neurons that are sensitive to cold, are activated and they cause sympathetic activity, which conserves heat. So I think that's pretty neat, to be honest. And then the prolactin and the hypothalamic nuclei. So the dopaminergic neurons are projecting from the arcuate nucleus in the hypothalamus into the anterior pituitary. That is the tuberoinfundibular pathway. And basically, dopamine is secreted by those neurons and acts on the lactotrophic cells via D2 receptors, and that inhibits prolactin formation. So dopamine inhibits prolactin, as we know, and that is why dopamine antagonists cause increased prolactin, and most people on antipsychotics have some degree of hyperprolactinemia. Then we've got the dopamine agonists such as bromocriptine, which decrease prolactin, and they're not really often used, to be honest, apart from in certain emergency situations. This is a bit of a random thing that comes up in this topic, which is, where does morality come from in a neuroscientific perspective, which is quite interesting, really. And I had a look into this, and basically the way that I understand it is that the ventromedial prefrontal cortex is the core of the emotional baseline that gives the instinctive moral intuition. So, for example, say you walk past a homeless person, you get that instinctive feeling to say, oh, let me give them all my money. But then the dorsolateral prefrontal cortex is the cognitive system that rationalizes it and says, like, don't give them all your money because you need to also get home and you need to buy your lunch. Why don't you just give them like £1 or something? So that basically kind of weighs up benefits and risks to behavior, cognitive control, and does problem solving. And it also is involved in lying. So yeah, just to remember that the ventromedial PFC, ventromedial prefrontal cortex, is that kind of like initial morality, the instinct, the kind of intuition towards like what you should do. And then the dorsolateral PFC is a kind of rationalizing tool. And that is quite, I think that's quite interesting, really, how that is developed. And I think there's different like theories of why that is, like as a social construct, but this is the neuroscientific basis for that. So now we are going to talk a bit about neurodevelopment and neuroplasticity. So in the syllabus, this focuses on kind of neurodevelopmental models, kind of development of the brain, that kind of thing. So let's talk about the embryology of the CNS. So this is the neurodevelopment of the brain. The CNS develops from the specialized region of ectoderm called the neuroectoderm. And the embryonic brain is divided into 3 areas, which are the forebrain, midbrain, and hindbrain. And then the forebrain is the prosencephalon. I think I should have a better picture of that here. So yeah, so the forebrain is the prosencephalon, the midbrain is the mesencephalon, and the hindbrain is the rhombencephalon, which is then divided into those 2 parts. So obviously these all make up the different parts of the brain eventually, and the prosencephalon separates into the diencephalon, which becomes the prethalamus, thalamus, hypothalamus, subthalamus, epithalamus, and pretectum, and the telencephalon. And also the telencephalon is what becomes a cerebrum, which is the kind of main cortex of the brain, the white matter, and also parts of the basal ganglia as well. So the main part is that cerebrum, which comes from the telencephalon. Then the mesencephalon is comprises the tectum, tegmentum, and cerebral peduncles, and a lot of the nuclei come from there. And then the rhombencephalon is like the kind of hindbrain, the bottom part of the brain, like going towards the brain stem, and that includes the medulla, pons, and cerebellum. And yeah, so just going back to that first picture, so this is what they become after those things, that forebrain, midbrain, and hindbrain. So I think in terms of essential knowledge for this bit, I'd probably just say to learn those 5 things: the telencephalon, diencephalon, mesencephalon, mesencephalon, and myelencephalon, and what they turn into. So the telencephalon, as said, that becomes a cerebral cortex, so the kind of main part of the cerebrum, lateral ventricles, and the basal ganglia. The diencephalon becomes the thalamus, hypothalamus, optic nerves, and the third ventricle. Mesencephalon becomes midbrain, cerebral aqueduct is kind of in the middle part. And then the mesencephalon becomes pons, cerebellum, and the last part of the fourth ventricle. And then the myelencephalon is the medulla, the bottom of the fourth ventricle. And then those are the 2 other things. So the alar plate develops into the sensory neurons, and the basal plate becomes the motor neurons. So the neurotrophins, neurotrophins are kind of part of the growth and development of neurons, anyway. So basically any kind of disturbances in these factors can help disrupt the creation of different kind of parts of the brain, different parts of growth, and it can be involved in schizophrenia and major depression. Typically, that's what's been studied. So there are increased amounts of BDNF, and that's actually a picture of what BDNF looks like, which is the brain-derived neurotrophic factor. So that's increased in cortical areas in schizophrenia, and it's decreased in the hippocampus, also in schizophrenia. Whereas in normal people, obviously that's kind of at normal levels, but those are the things that are reduced. And the neurotrophin 3 concentration, which is another neurotrophin, is reduced in frontal and parietal areas in schizophrenia. So yeah, just to remember that in schizophrenia, you get decreased BDNF in the hippocampus and increased BDNF in the cortex is probably the main point from that. And then talking about the cerebral cortex itself, so there's 2 main types of cells in the neocortex, which is basically 95% of the brain. That's the main area, that yellow part, what we consider to be the kind of like major aspect of the brain. And those are pyramidal cells and non-pyramidal cells. So with the non-pyramidal cells, there's 2 types, which are granular and stellate. And there's 2 types. One of them is excitatory glutamate, another one is inhibitory with GABA. I think we did talk about some of that before, but happy to go over it again at some point if needed. But yeah, the most important part of this is probably just to know the names. The archicortex, which is the oldest part, and that links the limbic system to the cortex. The paleocortex, which is in the olfactory areas, and the neocortex, which is the main part of the brain, made of those 6 layers, and that involves kind of pyramidal cells and non-pyramidal cells, which can have inhibitory and excitatory functions. And these are the Brodmann areas. So Brodmann divided up the brain into, well, divided up the cortex into these parts. And the most important ones to probably know are 44 and 45, which is Broca's, and then Wernicke's, which is 22. And I always just remember 44 and 22, and that Broca is 44 and Wernicke is 22, and it's half. So let's talk a bit about brain lesions. So again, this is a very big topic, and there can be a lot of different types of questions that can be asked from this section. And it can be a bit tricky as well to kind of just remember all of those details, because all of these words are so long and complicated. But let's start with the parietal lobe lesions. So parietal lobe lesions typically lead to apraxias, astereognosis, so when you can't tell what the thing is that you're touching, inferior homonymous quadrantanopia. I have some more things on visual field defects in a later slide, so we can talk about that in more detail. Gerstmann syndrome, which is usually in the dominant parietal lobe lesion, and it's classified by these 4 things. And again, I will touch on Gerstmann's again, but alexia, acalculia, finger agnosia, and right-left disorientation. These are typically the things found in parietal lobe lesions. And here, as mentioned, so this is Gerstmann syndrome. So it's a tetrad. So it's got 4 parts to it. And that's the kind of area that it affects. So it's also known as angular gyrus syndrome, because it's it's a lesion at the junction of the temporal and parietal lobes. So as you can see, that kind of junction there, that highlighted in yellow, is kind of between the 2, and that's the angular gyrus. So pure Gerstmann syndrome does not have aphasia. So if it has aphasia, then it can be classified as something else. But if you've got these 4 things, then you can classify it as Gerstmann syndrome. And basically, it's usually caused by a stroke in that area. They get finger agnosia, so can't tell like how many fingers someone's holding up. They can't write down, they can't do maths, they can't calculate things, and they get confused between right and left. So those are the 4 things. And yeah, so that's a pretty common thing to be asked. So worth just remembering those 4 things for Gerstmann's, and that is in the dominant parietal lobe. So now looking at the non-dominant parietal lobe, this has different kind of symptoms to it. Again, can be caused by a stroke or some other kind of lesion, like maybe specific like a brain tumor or particular thing, but it's probably more likely to be a stroke if you're getting specific symptoms. So you get the anosognosia, which is basically when they have a lack of awareness of a disability or a disease. Dressing apraxia, which is difficulty in getting dressed. Spatial neglect, lack of awareness of one side of the body, also known as hemisomatognosia. And then constructional apraxia, so they can't copy a picture of something or combine parts into something that makes sense. So those are the 4 things that I think are the most important. Again, you could classify that as a tetrad if you want for memory. So constructional apraxia, anosognosia, dressing apraxia, and spatial neglect. Now moving on to the occipital lobe lesions. So cortical blindness, and what that means is that the person has a visual defect, or they can't see, or they're blind, but it's not because of any problems at the front end, like everything's working fine in terms of visual apparatus at the front, but there is some reason in the cortex, particularly specifically the occipital lobe, that causes the blindness. So that's cortical blindness, so a lesion in the occipital lobe causing that. Visual agnosia, and you get homonymous hemianopia with macular sparing, and that is the key part of this. So the macular sparing is specific for occipital lobe lesions, and I'm going to just talk now a little bit about visual field defects as well, just to kind of bring that together really. So if you just go from the top down, so if we're going from the top, so number 1 is monocular vision loss, which is pretty straightforward. So you completely lose vision in one eye, and as you can see, that's obviously quite late down the pathway where number 1 is crossed over there. So that whole eye is messed. And then the second one is bitemporal hemianopia. So that's both sides that have vision loss. The kind of the right side of the left, no, the left side of the, okay, basically the sides are missing, obviously, that. We did this in medical school, but I feel like every time we have to learn it again for anything because it's impossible to remember. But yeah, that's usually at the optic chiasm. So yeah, that would be, I think we'll talk about that in a minute, but I'll just say it now. Actually, no, I'll save that for in a minute. But basically, it's the lesions of the optic chiasm, and depending if they're up or at the top or below, show you where the lesion is coming from. But we'll talk about that in a sec. And the next one's homonymous hemianopia. So both of the vision is missing on one side. So as you can see, maybe in number 3, that whole like purple tract, that whole side is missing. So you can see that innervates both of the right side of the eye. So both of those are missing. Then we've got the superior and the inferior homonymous quadrantinopias. So that's 4 and 5. And we'll talk about that in a sec. And then the last one, as said, so this is, in the occipital lobe, you get the macular sparing. So you may have whatever visual field, but the macula is generally spared if it's from that, if it's a lesion there. So yeah, and then the other thing to note is that there are congruous and incongruous defects. So when it's a congruous defect, it just means that there's this complete or symmetrical visual field loss, whereas it's an incongruous defect, it just means it's incomplete or asymmetric. So most of those in the examples are kind of congruous defects because they have a symmetrical formation, but they can also obviously be incongruous defects because of the nature of the way that the eyes work. And incongruous defects tend to be sort of more lesions of the optic tract, whereas congruous defects tend to be a bit further back because they tend to be more symmetrical if they're further back. And then the last thing that I was going to mention was with the bitemporal hemianopia, so number 2, that pink one, on the optic chiasm. So if the hemianopia is kind of focused more towards the upper quadrant, it kind of would indicate inferior chiasmal compression, and that's a pituitary tumor. And then in the lower quadrant, that would be a superior chiasmal compression, which is usually a craniopharyngioma. And as you remember probably from med school, it's kind of like switch round. So like if the lesion is at the top of the chiasm, it kind of causes the vision and the lower part of the vision to be missing, and then the other way around as well. So that can also be useful just to remember. And there's also the mnemonic for the homonymous quadrantinopias. So again, that is PITS, which is parietal inferior and temporal superior. So if you can see in numbers 4 and 5, so number 4 is like maybe a little bit closer towards the front of the person's face. So that would indicate, because that's a superior lesion, that would indicate temporal lobe, whereas the inferior lesion will be parietal. So the mnemonic is PITS, P-I-T-S. So parietal inferior, temporal superior, and that's related to those quadrantinopias. So 4 and 5, where you can see one's at the top mainly and one's at the bottom mainly, that relates to which lobe the lesion is in. Oh, I forgot that I did this slide, so that's useful. So yeah, again, so number 4 and number 5. So number 4 would actually be the temporal lobe lesion, which is a superior quadrantinopia, superior homonymous quadrantinopia. And then the number 5 would be the parietal one, because that's got an inferior. So both of the bottom left corners are blacked out. And then this looks like Illuminati. It's not, it was just me messing around, trying to make a triad. I think this is actually officially a triad for Balint syndrome, but basically there's kind of like an area in the kind of parietal occipital crossing over kind of region where if there's damage there bilaterally, it leads to this syndrome. So it's very rare, obviously, to get bilateral damage in both of those places, but that can happen due to atrophy, as you can see in that picture, or it can happen due to other things. So I had an example which was, so basically there's these 3 things. You've got the optic ataxia, oculomotor apraxia, and simultagnosia. So the oculomotor apraxia is the inability to voluntarily shift gaze despite intact function. So you can't look at something voluntarily, so you can't be like, oh, I want to look over there. That is sort of messed up by this syndrome. And then optic ataxia, sorry, that was oculomotor apraxia. And the second one is optic ataxia. So that is the disorder that affects the ability to interact with objects presented in the visual modality. So for example, if someone asks you to pick up a cup of tea, then you kind of overreach, or you can't see exactly where it is. And then simultagnosia, that's the lack of being able to perceive more than one object at once. So in a forest, or like in a car park, you could see like one car at a time, but you won't be able to see all of them. Or in a forest, you could see like one tree, but you couldn't like see all of them. So that's simultagnosia. So yeah, so the parietal occipital areas most consistently involved with patients with Balint syndrome are angular gyrus, the cuneus, and the precuneus. And as said, these bilateral lesions obviously are quite rare, but the etiologies are usually strokes. That's usually from transient global cerebral hypoperfusion. So if the person's like had an arrest and then they've had like loss of blood to their whole brain, and that's kind of reduced the, those border zones where the blood flow is least. That can cause that bilateral kind of reduced perfusion. Things like head trauma, primary and metastatic brain tumors, HIV encephalitis, progressive multifocal encephalopathy, and carbon monoxide poisoning can also cause it. But it can also be found in the neurodegenerative diseases such as Alzheimer's, Creutzfeldt-Jakob. And an example, so the lady whose scan that is, which has that bilateral atrophy that you can see at the bottom corners of the scan. So this was a 55-year-old lady who was evaluated for 5 years for progressively distorted vision. For example, she was unable to see two cars at an intersection on the road, and she had to be pulled by her husband because she stepped onto the curb as a car was approaching, because she couldn't see it. So that would be an example of kind of simultagnosia, or even also optic ataxia as well. So she could only see that one car, she wasn't able to see both of them, and that was quite dangerous for her. So I think with Balint syndrome, just the main thing to remember is the location, so kind of angular gyrus sort of area, the parietal occipital lobe region, and the fact that they can't kind of see more than one thing at once with simultagnosia. With the temporal lobe lesions, they can't recognize faces. They have auditory agnosia, kind of like can't like hear words, can't like understand what they mean, sort of thing. And they also get superior homonymous quadrantenopia, which is what we mentioned earlier with that Pitts thing. So temporal is superior, so they get that top corner quadrantenopia, and they also get the Wernicke's aphasia because Wernicke's area is in the temporal lobe. And then frontal lobe lesions, which are obviously the most sort of common lesions that we really see. Broca's aphasia, which is the expressive aphasia, and I always think of it as broken speech. So they can speak, but it's kind of non-fluent and it's broken, and it's not that easy to understand necessarily. They obviously have disinhibition, anosmia because the olfactory regions are in the frontal lobe, perseveration, and cannot generate a list. And this is the anatomy of the cerebellum, just before we go into kind of some of the cerebellar lesions. So essentially, the cerebellum, as you know, is primarily related to movement. It's got these two major hemispheres, like there's two parts of it, with the outer cortex, which is made up of grey matter, and the inside is made up of white matter. It provides the timing and the patterns of the skeletal muscle contraction for like coordination and moving around and your day-to-day things like opening the door, going upstairs, that kind of thing. We don't have any conscious knowledge of the cerebellar activity. Obviously, we don't have to kind of plan how it all works. And it compares where the body is in space and corrects any errors, which is quite amazing, really. Functionally, it's divided into 3 regions. So these are the anatomical regions: the flocculonodular lobe, which I think is a great name for a lobe, the posterior lobe, which is the kind of like main green section, and the anterior lobe. But in terms of functionality, it's divided into the 3 regions. So the anterior and posterior lobes divided into a medial section, which is the spinocerebellum, and a lateral section, which is the cerebrocerebellum, and then that has the vestibulocerebellum at the back, which is the flocculonodular lobe. So vestibulocerebellum does balance and spatial orientation, spinocerebellum does the fine-tuned body movement, and the cerebrocerebellum is involved in planning movement and the conscious assessment of movement. So yeah, that's quite a lot to remember, really. I can't say I remember many questions on the cerebellum, to be honest, but here we are. So this is a Danish pastry, because the mnemonic that most people remember from just general exams and med school is Danish. So cerebellar lesions classified by these things. So dysdiadochokinesia, which is that whatnot, ataxia, gait and truncal, nystagmus, intention tremor, slurred speech, and hypotonia. So that is Danish. Okay, it's just come up to 10:02. So should we just take a break till 3 and come back at 3 o'clock and continue there? If there's any questions, just pop them in the chat and I'll have a look at them after. Yeah. Okay, hi guys, welcome back. I hope you had a nice break. So what we've just been covering the lesions in the brain. Now we're going to talk about some cerebral asymmetry. So the planum temporale and Heschel's gyrus, these are the two kind of areas that come up where they talk about asymmetry. So the planum temporale is the portion of the cortical area of the surface of the posterior superior temporal gyrus, just posterior to the auditory cortex. It's known as Heschel's gyrus and is within the sylvian fissure. So that's the triangular region which forms the heart of Wernicke's area. And original studies found that the planum temporale was one of the most asymmetric areas in the brain, and it can be 10 times larger in the left cerebral hemisphere than the right. And that's usually in right-handed people, so their dominant hand. But then there can also be asymmetry in men just for no reason. So yeah, so the Heschel's gyrus and the planum temporale both exhibit some normal anatomical asymmetry. But however, in the planum temporale, in dyslexia and stuttering and in schizophrenia, there is reduced asymmetry. So that is something to note. The planum temporale has reduced asymmetry in those conditions. Then you've got the other lesions in the brain. So lesions of the mammillary bodies and medial thalamus for whatever reason. So can be stroke, can be alcohol related, can be kind of perfusion, whatever reason that can lead to sort of a Wernicke-Korsakoff picture. Any lesions in the basal ganglia in the subthalamic nucleus can cause hemimobilism, which is kind of like the movements of the limbs, which are a kind of like abnormal involuntary movements. Then you've got with the basal ganglia substantia nigra, you can have Parkinson's disease. Obviously, most people will be familiar with that one. And then basal ganglia striatum, so the caudate nucleus, that can lead to Huntington's chorea. And then in the amygdala, any lesions of the amygdala can cause Kluver-Bucy syndrome, which is a interesting syndrome that causes hypersexuality, hyperorality, hyperphagia, and visual agnosia. So these patients tend to put things in their mouth, eat a lot of food, have visual agnosia, and be hypersexual. So that is Kluver-Bucy syndrome, lesion in the amygdala. And now moving on to the blood supply of the brain. So again, something that we all probably covered in med school, but have to relearn every time for any exams. So as you all know, there's the three main supplies, which are the anterior cerebral artery, the middle cerebral artery, and the posterior cerebral artery. And these cover these areas. So the anterior covers, as you can see, the yellow area. So most of the kind of medial aspect of the cortex, the kind of inside top of the cortex, that kind of down the middle section. And then the middle cerebral artery kind of goes out to the side, goes to the temporal kind of region. And then you've got the posterior cerebral artery, which mainly goes towards the occipital region and kind of the back of the parietal region. So just talking a bit about some of those. So with the anterior cerebral artery, any kind of occlusions in the artery will cause a contralateral leg and foot hemiparesis and also sensory loss. As you may remember, the kind of the homunculus and that kind of sensory region is at the top of that cortex area. So that's all fueled by the anterior cerebral artery, as well as the bit of the motor part that does leg and foot. So that is affected. And then you've got some of these other more kind of specific ones, I suppose, which are the transcortical motor aphasias, where they can understand and repeat, but they're not fluent in the speech. And we'll talk about aphasias soon as well in the next coming slides. So we'll cover that in a moment. But the other things are that the primitive reflexes can emerge. So the frontal, what's known as frontal release signs. So kind of things like grasp, some of the other reflexes that are common in kind of primitive like development sort of thing. And then obviously you get the disinhibition, executive dysfunction, because all those kind of regions in the frontal lobe are responsible for those things. And obviously if you have a anterior cerebral artery occlusion, that will affect that as well, depending on where it is. And also they have impaired kind of insight into their own condition with the anosognosia. So yeah, you can see like in the image that the internal carotid supplies that ACA, does also branch into the MCA, but focusing here on the anterior cerebral, it goes sort of towards the front of the brain. It's supplying all those areas at the top in the cortex, and also kind of around the corpus callosum as well. So looking at that. Then looking at the middle cerebral artery. So as said, this also comes out of the internal carotid and it kind of branches out mainly to the temporal region. So when you're thinking about it in a 3D way, it's kind of makes a bit more sense really to imagine it coming out from the middle and kind of spreading outwards. This is the most common artery that's affected for strokes and generally. Any kind of occlusions in this will cause motor and speech issues. And with this, you'll get the contralateral face and limb hemiparesis with sensory loss. So you're kind of thinking more MCA if it's kind of face, arms, kind of that sort of thing, whereas you're thinking more ACA if it's foot, legs. Obviously, if it's both, then like could be a bit of both, but in exam questions, it might ask you more specifically in terms of the region. And then also there's there can be dysphasia or aphasia when the dominant hemisphere is affected, which has Broca's and Wernicke's region in, so that can also come up too. And then you get the homonymous hemianopia or quadrantanopia without macular sparing. So if you remember that with the occipital lobe lesions, there is macular sparing, so you kind of get that little gap for the macula, whereas when there's a homonymous hemianopia or quadrantanopia from a middle cerebral artery occlusion affecting the vision, you know, kind of an optic radiation part, that will have no macular sparing, so it will just be completely like, it won't be a complete straight line, but for exam purposes and theoretically, it would be like a straight line down the middle, so the eye's like black on one side. And then there's the dorsolateral prefrontal dysfunction as well. So the dorsolateral prefrontal cortex is involved in cognitive functions such as executive function, response selection, and working memory, and also in motor learning has been recently discovered also. And now the posterior cerebral artery. So yeah, so basically, as said before, this kind of is more coming up towards like the back of the brain, like the occipital region. You're having more visual field issues. This also supplies kind of like spinothalamic type things, so pain and temperature, sensation loss, contralaterally, prosopagnosia, so like can't recognize faces, same side cranial nerve defects, so that's ipsilateral cranial nerve defects, Horner syndrome, and then alexia without agraphia, which we'll discuss in a moment, but that is the left. Usually if the person's like right-handed, then that will be the left PCA. That makes sense. Then also you've got all those kind of other ones, you know, like lateral medullary syndrome. That's also known as Wallenberg syndrome, which occurs following inclusion of the posterior inferior cerebral artery, which cerebellar artery, which I call PICA. I don't know if that's what you're supposed to call it, but that some of those kind of posterior cerebral arteries can have those more specific ones like Wallenberg's or Weber's syndrome. lateral pontine syndrome as well. And then any infarct of the basilar artery causes something called locked-in syndrome, where the patient kind of is like paralyzed but has awareness. And then anything for the retinal or ophthalmic artery can cause amaurosis fugax. So this is just a summary to kind of look at overall what can happen. So as said, large vessels, so your ICA, that will obviously cause symptoms consistent with the MCA stroke or ACA stroke, because the ICA goes into the anterior and to the middle as well. Monocular blindness, which is amaurosis fugax, as said, if ophthalmic artery is affected. And then vertebro-basilar system, so that's kind of like at the back of the brain. I'll just show you that picture again. So yeah, so you can see like the anterior spinal artery, and then you've got the vertebral arteries, and that's going into the basilar artery. That's kind of like the back of the brain. So vertebro-basilar system can cause cranial, anything including that can cause cranial nerve palsies, diplopia, dysarthria, dysphagia, dizziness, vertigo, and any kind of those things there. And then looking at the medium vessels, you've got ACA, MCA, and PCA, which we've been through, but just a reminder that ACA is kind of motor and sensory deficit, which is more like legs rather than like face or upper limbs. And then MCA is more upper limbs and face. And then with the ACA, you get the primitive reflexes, from the frontal release signs. And then with the MCA, you can get aphasia if the dominant hemisphere is affected. That can affect the kind of language centers. And then neglect as well if the non-dominant hemisphere is affected. Then with the posterior cerebral artery, you've got the homonymous hemianopias, alexia, visual hallucinations, sensory loss, and then ipsilateral cranial nerve palsy, some motor deficits. And then you've got the small vessel, which are the lacuna ones, which I haven't really covered, but you can just look them up, sort of things like Wallenberg's, Weber's, and those things. So they can just be like kind of pure motor hemiparesis, pure sensory deficits, and kind of random little bits and pieces here and there. It can kind of affect anything, to be honest, because small vessels supply so many different parts of the brain that there's no necessarily particular syndromes that can occur. It can just be isolated issues or like general issues. And then this is just some of the gyri, which are really important. So the ridges in the brain. So the cingulate gyrus, this sometimes can be an exam topic. So the cingulate gyrus is kind of like cingulate means belt, so it's like a connection. It kind of wraps around the corpus callosum, and it basically is related to emotion, sensation, action. So it's part of the limbic system, process of emotion. So just remember, cingulate is emotion. That should get you through that bit. Angular gyrus is for language, maths, and cognition. Fusiform gyrus is for face recognition and also word and number recognition. So looking at kind of remembering like shapes and images. Precentral gyrus is for voluntary movement control. The postcentral gyrus is for touch. Lingual gyrus for dreaming and also word recognition, but that's quite interesting, lingual dreaming. Superior frontal is for self-awareness and laughter. Superior temporal is related to language, and it has reduced volume in schizophrenia. And then the parahippocampal gyrus is involved in memory, and there is asymmetry in schizophrenia. And then the dentate gyrus is part of the hippocampus and is involved in the formation of episodic memory. So yeah, I think I did. Yeah, so I mentioned the fusiform gyrus. So with that does face recognition, but so the term for that is prosopagnosia, which is face blindness. So if the question asks which area does prosopagnosia, the answer would be fusiform gyrus occlusion or lesion. Okay. And these are some of the behaviors that can come about from various different reasons, like They can be from lesions, or they can be kind of from like different areas being occluded, tumors, whatever reason there are. But these are kind of some of the behaviors that come out of that. So utilization behavior is using objects appropriately but in an inappropriate setting. So they usually will have the object there and then use the object in the correct way that the object's supposed to be used, but the setting is not appropriate. So like you're sitting on the train and you give someone like a toothbrush, then they just immediately start like brushing their teeth. Like they're doing the right thing with the toothbrush, but it's not really where you should be doing it. And again, like brushing your hair, at the doctor's or whatever. So yeah, these are all kind of in the frontal lobe because they relate to executive function and kind of social behavior. And then imitation behavior. So this is when the patient imitates the examiner. So while the person is talking, like whatever like movements they're making, they start like copying what they're doing, or like if the person like scratches their head, they stop doing that. And then alien hand sign. So the patient feels like they have no sense of control over their movements, which is, I think, is different, can be distinguished from passivity phenomena, because they don't feel that somebody's controlling their hand for them, but they just feel that they don't have any control of their hand and it's doing kind of whatever it wants, really. And then manual groping behavior, so hands kind of, or eyes watching kind of follow objects. Grasp reflex is a primitive reflex, so the automatic behavior of gripping objects in hands. I'm sure like most people have had this before, like for example in elderly care, with very severe dementia, like if you put your hand into like someone who has that and into their hand, they kind of grasp onto your hand and don't let go of it. That is grasp reflex. And then environmental dependency syndrome. So they rely on the environment to guide what is an appropriate behavior. So if you take them to a restaurant, then they will know like that they should be eating. Or, if you say, oh, we're currently on a cruise ship, then they might start looking out the window and saying, oh yeah, look, the sea is very lovely, or whatever. So they're kind of dependent on what their environment looks like or what they're told about their environment, and that tells them like how they should be behaving at a particular time. I can't say that most of these things sound like they're very common. I don't think I've seen like any of this stuff really before. I don't know if you guys have. Let me know if you have. I'd be interested to know your own experiences if you've seen any of these in clinical practice or come across them. I think the only one I've probably seen before is grasp reflex, but the other ones, or maybe like manual groping behavior, but that's kind of a bit neither here nor there really, because eyes following objects are not necessarily that strange. So let's talk a little bit now about aphasias, apraxias, and agnosias. So from your vision, you may know that there's two types really. There's the perisylvian aphasias and the extra-sylvian aphasias. And the perisylvian aphasias are mainly centered around this lateral sulcus, which is the fissure of Sylvius. So the Sylvian fissure is basically kind of around that region. You've got all of these language areas. So you've got Broca's area and Wernicke's area, and between them you've got the arcuate fasciculus, which connects them. So that's known as the perisylvian language area. We'll go through each of those. So with global aphasia, this is the most severe version of the aphasias. They're completely non-fluent, they can't understand, and they also can't repeat as well. So that would be a lesion of that entire perisylvian region, including Broca's and Wernicke's. This is on the left, by the way, in most people, because most people dominant, like right-handed people, would have left their Broca's and Wernicke's on the left. And this is usually from an MCA occlusion, global aphasia. So you're thinking, if you remember that picture of the way that the blood flow from the MCA supplies all of that temporal region, that whole area is supplied by MCA pretty much. Obviously, there's part of Broca's that can be like dually supplied by the anterior cerebral artery, but it's usually MCA occlusion that will cause a global aphasia. Then you've got the Broca's aphasia, which is damage to Broca's area 44 and 45 in the frontal lobe. So this is, as I said before, I remember it as brokers is broken speech. So when someone's got broken speech, you can still understand what they're saying, but it just doesn't sound as fluent. So that's the way that I remember it. So they can speak and the understanding is intact, but they do have a bit of disordered repetition. So, that part of the arcuate fasciculus that is a bit messed up, really. And they also can't write. So this is usually the superior division of the MCA that causes Broca's aphasia from a blood supply point of view. So Wernicke's aphasia, this is fluent but meaningless speech. So they basically speak nonsense. It kind of sounds like it should make sense, but it doesn't really, but it's quite fluent. So this is also known as cocktail party speech. Basically, you go to a party and someone's talking nonsense. And this is the inferior division of the MCA. And they also can't understand what other people are saying as well. And then conduction aphasia, see that link between the two of them. So you've got the arcuate fasciculus there. Any damage to that disrupts repetition. And that can be the MCA inferior or superior divisions or both. So kind of anywhere along that line, if there is any disruption to the blood flow, to the arcuate fasciculus, that will interrupt repetition and ability to kind of repeat things. And then we've got the extra-sylvian aphasia. So we've already mentioned the perisylvian ones. Those are repetition impaired because they're using the arcuate fasciculus, which is the link between those two. But with the extra-sylvian ones, the repetition is preserved because there isn't really damage to that arcuate fasciculus. With these extra-sylvian aphasias, they're kind of outside of the core perisylvian language zone. So they occur less frequently, and they may also arise from infarct, but they can be affecting other areas. So with these ones, you've got the anomic aphasia, which is naming or word finding difficulties. That's the main feature. The grammar is not affected and repetition is good, and also auditory understanding is fine. And anomic aphasia is very difficult to localize because it can be sort of in any area, but it's often in the temporal parietal area around this language zone, but not necessarily any particular place. And it can also be in the angular gyrus area as well. With the transcortical motor aphasia, so spontaneous output is disrupted, and they're not very fluent, and they kind of have a kind of stuttering as well, but they can repeat everything perfectly. So they can repeat whole sentences like we would be able to normally without any kind of difficulty, and comprehension, so understanding is fine, and naming may be a little bit impaired, but it's otherwise okay. And then transcortical sensory aphasia is basically the same as Wernicke's, so they're kind of speaking fluently, but it's not making sense, and they can't understand, but the repetition is preserved. So transcortical sensory aphasia is basically Wernicke's, but with repetition preserved. And then the subcortical aphasia. So these are focal lesions specifically on certain subcortical structures, such as like the thalamus or the striatum, and they're usually related to kind of like tumors in specific areas, or like abscesses, hemorrhages, specific lesions. But yeah, that's basically subcortical aphasias. And then this is just a summary of kind of how those fit, really. So when we say an aphasia, we say like, are they fluent or not? So if they are fluent, then it could be any of those. So you've got the two Wernicke's or transcortical. So the difference between Wernicke's and transcortical sensory is that in transcortical sensory, they can repeat, but in Wernicke's, they can't. And then the other type of aphasia on that side, so the conduction and the anomic. So yeah, so they have ability to kind of say the things fluently, and comprehend them, not comprehend them. Wait, comprehend them? No. And then with the repetition, they either have yes or no for that. And then on the other side, you've got Broca's with the transcortical motor aphasia. So that's a kind of version of Broca's that can repeat. So if you think Broca's and Wernicke's and then motor and sensory, so they kind of have a pairing there. And then with the other ones, you've got the global aphasia. That's basically everything is trashed. And then the mixed cortical, transcortical aphasia, which can be kind of a mixture of various different things. So yeah, I found that quite useful. Obviously, it's a bit hard to remember it off by heart, but if you just maybe go through some of the questions kind of online related to these, that should be able to solidify that in the memory. And then the other syndromes are, yeah, these always, I always got very, very confused with these. So basically, alexia without agraphia, that is pure alexia. So basically with that one, the person who normally can read and write presents with an acquired loss of reading ability. So they stop being able to read, but they can write. And then oftentimes the example given is that a person has written something down themselves, and then you ask them later to read it, the same thing that they've written, and they can't read it back. So that's pure alexia. And speech is normal in that one, and the main thing is the angular gyrus that is affected, and causes include left PCA, occlusion, MS, kind of tumours as well. And then alexia with agraphia, so this is basically worse than the first one because again it's angular gyrus, but they can't read or write anymore, and usually this is in people who have previously been literate. So yeah, that happens, but then they have very good preserved oral language functions. So their speech is fine, although they sometimes may have like word finding difficulty, but auditory comprehension and repetition are intact. So with alexia with agraphia, it's just a very complex way of saying can't read and write anymore. And then pure word deafness is similar to Wernicke's aphasia, as understanding is impaired, but speech is fluent, but they can understand kind of meaningful sounds like beeping cars or like telephone rings. Which I don't really know how that looks in person really clinically. I mean, it must be quite specific really. But yeah, they don't have, so they can't comprehend like word sounds, but yeah, they can comprehend other meaningful non-verbal sounds. And these are some of the agnosias. So we've mentioned some of these already as we're going along, but it's always just helpful just to remember what they are. So anosognosia, so the person doesn't have recognition into their own illness, their own disability, or any kind of thing like that. I think it's quite similar to insight, I guess, but maybe a different kind of way of expressing it in a way, because they cannot recognize their own condition, and it can also be physical things, you know, like their visual loss or other things like that. Autotopagnosia, so they can't orient parts of the body. Phonagnosia, they can't remember. They can't recognize familiar voices. Simultagnosia, and that's can't appreciate two objects in the visual field concurrently. So if you remember in Balint syndrome, that lady who nearly got hit by the car couldn't see that there was another car there. So that is why, you know, that's a good way to remember simultagnosia. And then asteriognosia, which is people can't recognize things by touch. Then we have the visual agnosias. So these are kind of familiar objects that you may know what they are but can't recognize them, but there's no problem. The key thing is that there's no actual issue physically with the sensory parts, like the eye itself is completely intact. and the visual kind of systems are fine, but the person just can't necessarily see certain things. Prosopagnosia, can't identify faces, and we mentioned before that's often related to the fusiform gyrus. And then simultagnosia, we've also mentioned before, can't recognize a whole image, but details are recognized, or they may be able to see one thing at a time, but not more than one. So Anton Babinski syndrome is another one of the kind of specific syndromes that can be asked about. So they kind of had this confabulation of what they describe as their environment. So they have blindness from the cortex, so cortical blindness. So this is blindness caused by a lesion in the occipital lobe. But they don't know that they have this problem, so they have anosognosia of that. So they have no insight or no appreciation for the fact that they have this blindness. They often fall over things, but they believe that their vision is fine, and they describe their environment. So they say, yeah, the table's there, the chair's there, but it's actually not true. And they believe that that is what they are seeing, and is incorrect. So they tend to fall over things. So that's Anton Babinski syndrome. Again, quite interesting. If any of you guys have ever seen this, I'd love to know, because obviously we study this, and I don't think I've ever heard of anyone that's met anyone who's had this before. So yeah, do drop me a message if you have. So then the apraxias, again, these are like classic questions. I got this in both of my papers that I did both times. Often the one that is asked is the difference, not the difference, but they will give the two options, which are the ideomotor apraxia and the ideational apraxia, and then try and get you to kind of like say which one it is. And with ideomotor apraxia, the object is used for wrong task. So you give someone, you want someone to write something down with a pen, and they start like using it to comb their hair or something. And then ideational is like you can't follow sequence of action. So I put a cup of tea there. So they wouldn't be able to make a cup of tea. That's ideational apraxia. So then going back to the top, so limb kinetic apraxia, they can't make fine movements. Constructional apraxia is that they cannot copy a picture or combine parts together. And then oculomotor apraxia is that they cannot control eye movements. So now we're going to talk a bit about neurodegeneration, which is 3.10 in the syllabus. Covers some of these things like Alzheimer's, Pick's disease, Lewy Body, prion. So we'll just go straight into that. So with Alzheimer's dementia, you've got obviously the macroscopic changes that you see in the dementia. So cortical atrophy, which is usually with occipital sparing. So usually affects the kind of frontal, temporal, parietal lobes mainly. So mainly temporal, medial temporal region actually. You get widening of the sulci, ventricular dilatation, depigmentation of the locus coeruleus, but the substantia nigra is preserved, which is a key point to note, because in Parkinson's, you get depigmentation of locus coeruleus and substantia nigra, but just remember that in Alzheimer's you get depigmentation of locus coeruleus, but not of the substantia nigra, and that's again why we use the acetylcholinesterase inhibitors to try and bring back some of the acetylcholine that's lost from like that degeneration of the locus coeruleus, which is a site for synthesis of acetylcholine. And then there's also reduced brain weight as well in Alzheimer's dementia. So yeah, cortical atrophy, shrinkage of the cortical gyri. This is predominantly of the hippocampus, parahippocampal gyrus, and temporal amygdala. And then yeah, so that's kind of the main parts. And then as said, next is parietal and frontal lobes most severely affected, and the occipital lobe is generally spared. So that is in Alzheimer's. And then macroscopic changes. So these are changes that occur mainly obviously in the cortex. You get beta amyloid deposits, and this is from the APP protein, which is the amyloid precursor protein. You get beta amyloid from that, from the breakdown of that. I'll mention that later as well about how that's made. And then also you get neurofibrillary tangles made from hyperphosphorylated tau protein, gliosis near the amyloid plaques, and degeneration of the basal nucleus of Maynard. Again, site for, as we said before, and Hirano bodies, so eosinophilic actin-rich intracytoplasmic inclusions. So these come up as pink on the with eosin, and they're usually found in the hippocampi as well. So that is Hirano bodies. And then there's an important thing to note about Hirano bodies, which are that they are very similar to Lewy bodies, but one thing is that Lewy bodies contain ubiquitin, whereas Hirano bodies don't. So yeah, I just want to check one thing actually, because yeah, I think I said wrongly that about the locus coeruleus, because that's for noradrenaline, and in Alzheimer's you get depigmentation of the locus coeruleus, which is the blue spot producing noradrenaline. But the main thing is actually the degeneration of the basal nucleus of Maynard, which is where the acetylcholine is produced, and that's why you need acetylcholinesterase inhibitors. So sorry about that, I made a mistake there. But otherwise, apart from that, the main thing to note is probably Hirano bodies don't have ubiquitin. You get degeneration of that basal nucleus of Maynard. You get beta amyloid deposits and neurofibrillary tangles, and gliosis as well. So there's kind of multiple reasons why Alzheimer's dementia is quite bad and difficult to treat, because of all those microscopic changes. And so when you're trying to distinguish between Alzheimer's dementia and dementia with Lewy body, there is a way to discriminate between the two using SPECT scans. So dementia with Lewy body has lower perfusion in the occipital cortex, and Alzheimer's dementia has low perfusion in the medial temporal areas. So that's one way to tell the difference between the dementias. If you want to send them for a SPECT scan, you'd be able to tell what the difference is there. So tauopathies. So these are kind of the diseases caused by tau protein build up in certain ways, and these neurofibrillary tangles are formed by this hyperphosphorylated tau. They're found in frontotemporal dementia, the progressive supranuclear palsy, corticobasal syndrome, and dementia pugilistica. And with dementia pugilistica, that's the also known as that kind of punch drunk syndrome the boxers get, where they have a form of dementia seen in repeated head injuries, usually in kind of fighters or boxers. It's also known as chronic traumatic encephalopathy, and it's a neurodegenerative condition. And symptoms can occur after just one brain injury, but it's usually after chronic. And with that, you can get slurred speech, hearing problems, neurobehavioural disturbances, cognitive decline, gait ataxia, and they also get other kind of behavioural things like aggression, suspiciousness, childishness, depression. It's hard to tell like whether that was just the person's personality, in my opinion, but I suppose if they're having kind of things like gait ataxia and things, then that could be relevant. And the other thing is that one of the key words for that is the septum pellucidum. So kind of any damage to the septum pellucidum, you're thinking dementia pugilistica. That's the key word there. Yeah. Oh yeah, so that you also get tau build up in the depths of cortical sulci in that type. The frontotemporal dementia. So there's actually 3 kind of syndromes that come under this, and frontotemporal dementia is an umbrella for those 3. And we'll touch on this more in molecular genetics because it's got a lot of kind of genetic component to it. But essentially, Pick's disease, which is what we sometimes say, is kind of known as now behavioural variant frontotemporal dementia, and is related to the MAPT gene, and is caused by a 3R tauopathy. Again, we'll go through some of this later, so don't worry too much about it. It's good just to know the keywords, I guess, but just to be aware that that's what Pick's disease is. Then you've got the semantic variant PPA and non-fluent variant PPA. And those 2, so semantic and non-fluent, involve the genes GRN, C9ORF72, which I did actually get asked in my second exam, I think, VCP and TARDBP. Don't ask me about the last 2 as I can't remember what they are. And the key thing that is about FTD is they have this knife blade appearance. So there's focal gyral atrophy. So it's like a knife has been cut through them. They're really, really deep ridges and sulci with the FTDs. So it overlaps with 3 other neurodegenerative diseases as well, which is the corticobasal degeneration, progressive supranuclear palsy, and amyotrophic lateral sclerosis as well. I think maybe we'll look at more of that in classification. So going on to dementia with Lewy body, that's a Lewy body. It's basically alpha-synuclein, and it's an intracellular protein accumulation. And as said before, the difference between a Lewy body and Hirano body is that the Lewy body also contains ubiquitin, which is a protein. And in dementia with Lewy body, there is pallor of the substantia nigra and the locus coeruleus. So that will go with the Parkinson's type of picture. And as said before, when I incorrectly said about acetylcholine, that's from the basal nucleus of Mena, and that's in Alzheimer's disease. There is also depigmentation of the locus coeruleus in Alzheimer's, but preservation of the substantia nigra. But in this one, the dementia with Lewy body, you've got pallor of both substantia nigra and locus coeruleus as well. So yeah, cerebral atrophy, less than Alzheimer's, pallor of both. You got the Lewy bodies with the ubiquitin and alpha-synuclein, but you do also, which is same as Alzheimer's, have the neurofibrillary tangles and the senile plaques present. So this is about alpha secretase, which is the enzyme, which is it's kind of a focus now of how they're trying to develop medications to improve, a lot of these neurodegenerative disorders, because they're trying to develop drugs that stimulate alpha secretase or block beta or gamma secretase. So APP, which is amyloid precursor protein, it crosses the cell membrane, then it gets broken down by alpha secretase into non-toxic fragments. But if gamma or beta secretase break down APP, it breaks it into these short sticky fragments, which is known as beta amyloid, and that's what joins together to produce insoluble amyloid plaques. So alpha secretase is a good one, so I think like alpha is the best, and then gamma and beta not so good. Like they cause this beta amyloid, these short fragments to be produced, which then stick together and cause these insoluble amyloid plaques, which cause neurodegenerative processes. Prion disease. This can be, as mainly the questions are related to Creutzfeldt-Jakob. So that can be, so prion diseases can be, they can be from infected meat products, so that can be pork or beef usually, or can be other animals too. So with the main questions being about CJD, that the focus is on the differentiation between variant CJD and classic CJD. So the difference is that with variant CJD, this is the one that we get, or hopefully not we get, hopefully we don't get it. This is the one that people get from eating infected meat products, and it usually affects people in their 20s to 30s. It takes a long time, more than a year usually, presenting with the psychiatric symptoms first and neurological symptoms later. And it's classified by this pulvinar sign on the MRI, which often comes up in question banks, and the generalized slowing on the EEG. Whereas in classic CJD, which is a sporadic CJD, this is caused by genetics, so not by infected meat. It's got a shorter duration, so it comes on after like months. It presents like either the same time with neurological and psychiatric symptoms, or just first of all with neurological symptoms purely. It's got a high signal in basal ganglia, bilateral anterior area, but it's not the same as the pulvinar sign. And there are biphasic and triphasic waves on EEG. And it affects an older age group of around 55 to 65 years old. Okay, let's see the next one. And then Parkinson's disease. So obviously everyone remembers Muhammad Ali, who had developed Parkinson's disease. The pathology is similar to dementia with Lewy body. So there is that pallor of the substantia nigra and the locus coeruleus, which is the same. And again, as said, so you're not getting the pallor in Alzheimer's of the substantia nigra, only in the locus coeruleus. And then in the microscopic changes, the substantia nigra pars compacta is losing dopaminergic cells, and that is, part of the low dopamine thing, plus Lewy bodies. And then also the other thing with the difference between Parkinson's and dementia with Lewy body is that with the Parkinson's, you're getting the kind of physical manifestations first. So kind of like the gait, shuffling gait, those kind of things, the tremor, that's all coming up first before the more like neurological, not neurological, like psychological kind of like behavioural stuff that comes later. Whereas dementia with Lewy body, you're getting that kind of the mind stuff first, and then later you're getting maybe some of the more physical stuff coming on with it. And then these are some like relevant pathology notes. So like Kuru plaques, which is a prion disease, that's present in Kuru and Gerstmann and Straussler, and also sometimes CJD. It's a type of prion protein. Hirano bodies are the eosinophilic rod-shaped, non-ubiquitin things found in Alzheimer's dementia. Neurofibrillary tangles are in Alzheimer's dementia, made of microtubular proteins. Then you've got your zebra bodies, which are found in Niemann-Pick and Tay-Sachs, and also your other like mucopolysaccharide diseases. Bar bodies are the inactivated X chromosome. Asteroid bodies are stellate inclusions in giant cells, which are found in sarcoid and berylliosis. And then Pick bodies are the intracytoplasmic spherical tau fibrils. Pap lantos found in multisystem atrophy, glial inclusion bodies, and then Lewy bodies are found both in dementia with Lewy body and Parkinson's disease, and they're alpha-synuclein intraneuronal inclusions. And what I would say about this is obviously it's a bit long, but oftentimes the questions will ask about like if it's intraneuronal or intracytoplasmic, or kind of like the bit that you think is not important about the about this part is sometimes what they ask. So it's worth kind of knowing like which part they go to or they work in terms of inside the cell, inside the neuron, and whether extracytoplasmic or intracytoplasmic. Usually they're intracytoplasmic in these, but just having a look through that and revising that as well. Then we've got the hydrocephalus. So this is classified as a reversible cause of dementia. It's caused by reduced CSF absorption at the arachnoid villi, which means that the CSF builds up in the brain. It's a classic triad of urinary incontinence, dementia, and gait ataxia. It's a little triangle, so you can just remember that. And the treatment for that is a VP shunt, so just to get that fluid moving down, and that is the treatment. I don't know how often again. I think that this is probably one of the more common things that we may see from time to time, but again, not that common. Motor neuron disease. So this has got an unknown cause to it. So see, yeah, so it's a progressive neurodegenerative disease. It attacks the upper and lower motor neurons. Degeneration of the motor neurons leads to weakness and wasting of the muscles, difficulties with speech, swallowing, breathing, loss of mobility in the limbs. The diagnosis is clinical diagnosis, but you can do nerve conduction studies, and they show normal motor conduction. And then EMG, so electromyography, shows reduced number of action potentials with increased amplitude. And then MRI is also usually performed to exclude cervical cord compression or myelopathy. And the key thing to note is that they never, so probably not never, but for exam purposes, they never have sensory symptoms. So they may have like some vague sensory symptoms in the early course of their disease, like limb pain, but they don't. This is normally just pure like motor symptoms. Yeah, so like mixed upper and lower motor symptoms, and not sensory symptoms, because it's only working on the motor neuron. So that makes sense, right? And there's actually the 4 types of motor neuron disease. So you've got amyotrophic lateral sclerosis, which is upper and lower motor neuron involvement characterized by limb weakness. Progressive bulbar palsy, which is upper and lower, again, neurons, but it's characterized by speech and swallowing problems. Progressive muscular atrophy, which is mainly lower motor neuron involvement, characterized by weakness and clumsiness. And then primary lateral sclerosis, which is only upper motor neuron involvement and is mainly characterized by lower limb weakness as well. So yeah, so those are the main 4 types of MND. And then some of the pathological features, macroscopic and microscopic. So macroscopically, you've got atrophic anterior nerve roots, spinal cord thinning, frontotemporal atrophy, and the precentral gyrus atrophy. Then microscopically, you've got a loss of motor neurons from the lower brainstem and loss of motor neurons from the ventral horn of the spinal cord. So obviously the neurons are microscopic, so the microscopic features are mainly just focused on the motor neurons in the brainstem and the ventral horn of the spinal cord. And then macroscopically, you can see that kind of atrophy of the anterior nerve root. The spinal cord appears thinner, and you've got some frontotemporal atrophy and precentral gyrus atrophy. And just a reminder of the things in the upper motor neuron lesions. So you've got weakness. I always think of these like all up. I think most people do, like upper motor neuron lesion, everything goes up. So you get upgoing planters, increased tone, clonus, like reflexes are brisk. You get mild atrophy, and you do get weakness. But then in the lower ones, you get more atrophy. You get decreased reflexes, decreased tone, weakness, and fasciculation as well. So I think that brings us sort of to the end, really. I was just going to say that I've put a note here that we haven't covered epilepsy yet, and some of the other things, but most of those we'll be covering in later sessions, going over some of the classifications of diseases. Epilepsy will probably come up under that. And again, as I said before, I tried to cover everything that I thought was most important, but obviously there will be some things that you'll find in questions that may not have come up today. So yeah, like if you have any feedback or you want to add anything, then do let me know. But otherwise, we'll take questions. I've got some questions already, so I'll just start having a look at those, and then yeah, okay. So one of the questions was, can we repeat the visual field defects and sites? So yeah, we can do that. Where are they? Okay, there they are. Yep, so we've got, these are the kind of main ones. So you've got, going from the top, so from the front of the eye, you've got monocular vision loss. So one eye is like you can't see out of one eye, and that is usually from that lower down in the tract. So after the optic chiasm, any kind of defect there, and any injury there, or any kind of demyelination at that point will cause a monocular vision loss. Then you can see bitemporal hemianopia. So that's at the optic chiasm right in the middle where they cross over. That's where you find your pituitary adenomas, craniopharyngiomas, and that is the optic chiasm. So that's a bitemporal hemianopia. So both on the outsides. And I will go to the other slide that shows about the well, maybe I'll just do it now. No, not that one. Yeah, so here we go. So in the optic chiasm, if it's more in the upper quadrant that the vision loss is, then there's pituitary tumor and the kind of this inferior chiasmal compression. Whereas if it's more in the lower quadrant, then there's superior chiasmal compression. Obviously, it's like hard to tell because on that picture, it looks like that whole side is obviously blacked out. So, but that is just kind of like one of the notes that is made. And then the next part is the third bit, which is a homonymous hemianopia. So that's like both the same side. So both your right side or both your left sides can't see. And that's because of one side. As you can see, like, for example, the purple, if that was cut across, that would affect both the right side of both eyes. And then you've got the superior homonymous quadrantinopia and the inferior homonymous quadrantinopia. So this is from later down in that optic kind of radiation area, when it's going through the lobes towards the occipital lobe. If there's any lesions along the way when it's getting to the eye, or from the eye, whichever way around, no, to the eye. So no, from the eye. Yeah, be from the eye. You don't blast things out of your eyes, do you? So yeah, basically, if you've got a parietal lobe lesion, so that's the back one, then that will cause an inferior homonymous quadrantinopia. So the bottom left-hand corner of the eye would be missing. If it's in the parietal lobe, that's a bit further back. And then the one that's further forward, to number 4, that causes, if it's in the parietal lobe, wait, did I say that already? Yeah, no, that'd be in the temporal lobe. That causes superior quadrantinopia. And then that's that mnemonic is PITS. So parietal inferior, temporal superior. So just remember that for homonymous quadrantinopias. And then the last one is the macular sparing. So this is, just occipital lesion causing, like just right at the back, causing damage to that whole side of the eye, of the tract that goes to the eye, and it causes macular sparing. So I hope that helps. Let me know if there's any other questions. So the other question was, what is the meaning of hypoorality? So it's just kind of excessive, like putting things in the mouth, like chewing, biting, kind of swallowing things, not necessarily eating them, because that would be like hyperphagia, but hypoorality is just like putting things in the mouth a lot. What is the difference between Hirano and Lewy bodies? Where are they? Yeah, so just get that notes up. So Hirano bodies are similar to Lewy bodies. They are both intracytoplasmic inclusions, but Hirano bodies stain pink with eosin, and they usually they don't contain ubiquitin, whereas the that was it. The Lewy body is formed of the alpha-synuclein and contains ubiquitin as well. So, yeah, if that makes sense to you. What is the gold standard to diagnose Korsakoff? I think it might be. I think it's MRI of mammillary bodies, but I'll check that and get back to you. So I will get back to you on that one. Are there any other questions? Let me just check that. Thank you everyone for your nice comments. Appreciate that. I'm very glad that you're finding it useful. Also, I took my inhaler at the beginning, which did help with the not coughing. So I think I've worked it out finally now. Yeah, let me get back to you about the gold standard for Korsakoff. I'm not sure. I just had a look and it says there's not necessarily any imaging procedures that can be done, but I'm pretty sure that MRI of mammillary bodies was something that was mentioned somewhere. If anyone happens to know, then do message me. Otherwise, I'll get back to you on that one. Yeah, so I've got time now for if there's any other questions. Otherwise, if you guys are happy, then we'll, as said, have a break next week and then regroup the following week and take it from there. So yeah, just do your questions on basic neurosciences, just really hammer that down and get it all finished. And we will come back next time. I'll hang around for a little bit because there's some other questions, or if people want to ask me questions privately, that's fine as well. So yeah, thanks guys, thanks for listening. Hope you did find it useful. Glad that some of you said that it's been really good so far, so I'm very happy about that. But yeah, any feedback always welcome, as said before. Yeah, so resources I recommend for neurosciences. So I think I mentioned at the beginning, but I used MRCPsych Mentor. I would just recommend getting that. It's not too expensive. Just getting that and just doing the whole question bank. I did it twice. Well, obviously because I sat it twice, the exam, so I did it twice. But I think I tried to do it like 2 times. The second time I sat the exam, I think I tried to do the whole bank twice, and looking and going like through specifically. So, for example, just clicking on basic neurosciences and just doing those whole sections and doing that. And then I also used the SPMM mocks. I think that those two together probably were the main passing things for me. I think the first time around when I sat it, I did do MRCPsych Mentor questions, but I only did it once going through the bank, and I didn't do that SPMM mocks. Whereas the second time I did the bank twice and did the mocks. That was the main difference, I think, in terms of questions, if you're asking. To be honest, I didn't really read that much either time. I'm not really that good at reading notes. I prefer to have it like in this kind of format. That's why I obviously mentioned that that's why I'm passionate about this kind of thing, because I really want to create like a more interactive and useful place to learn. That's not just reading yourself off notes. But yeah, I hardly read any notes because I can't concentrate on reading notes. So I mainly just did those 2 banks, SPM mocks, and just watch some YouTube videos sometimes if there was anything I didn't really get. I did try to write some notes, but I didn't get that far. But I am using the notes that I wrote from gathering all of that info to help with these presentations, and I'm just using other resources that were useful and exam related stuff to put this together. I actually think I feel like I'm doing more work like getting these presentations together. Well, most of them I have done kind of like near when I did the exam, so it does help. But going through them again so that I can present them to you, I feel like I'm doing more revision now than I did for the exam. So anyway, I'm confident that you guys are going to do well. Some of you are sitting it in April, some of you are sitting it in July. Either way, I think you've got enough time to pull it together and like be confident. Like feel good and be happy. You know, it's gonna be fine. And the worst that happens is, you know, if you have to do it again, like I did, it's not the end of the world. It's actually all right. So yeah, if you guys haven't got any more questions, then like feel free to go. As I said, I'll just hang around for a few more minutes and then I'll go. And yeah, sometimes people do send me some questions later on, so I will just wait to see. Okay, thank you. I'm very glad. So someone said that it's a very helpful session. Good, thank you very much. Yeah. Okay. Oh, you're welcome, guys. I'm glad you like it, and have a good week. And I'll see you not next week, but the one after. So yeah. All right then. I'm gonna. Oh. I'm gonna end the webinar. So any other messages, just drop me an email or something. All right, bye everyone.